questions at specific times. And also we have these handouts that are little questionnaires for how you guys thought the talk went, if there's anything we can improve on. As Dana mentioned, we're also trying to train graduate students on how to communicate their science, so we really use this feedback to help our students get better at bringing these events to you guys. So that, I think we'll let Kelsey take it away.
All right. All right, thanks guys. As I said, my name is Kelsey, and I'm a 5th-year graduate student in the genetics department over at the medical school campus, but I work in a lab that studies neuroscience. And so today I'm going to tell you a little bit about the neurosciences related to my research, and hopefully you'll learn a little bit more about how your brain works. And so to start the talk, I want you to think about your day so far. So you woke up, you opened the curtains, then you were hungry, so you ate breakfast, and then maybe you got dressed, you went outside to go to work, and actually you can stop thinking about your day now because we've only been through the first maybe few hours of your day, and I've already made my point, which is that during your day you go through lots and lots of changes. So when you open the curtains, you experience a change in the light. When you had, say, Fruit Loops for breakfast, you had a really big change in the amount of sugar that was in your body. And when you went outside, it was a little bit colder than it was inside, so you experienced a change in temperature. But even though you experienced all of these changes, your body adapted to them, right? So when you experienced a change in light, you weren't permanently blinded by that light. When you ate breakfast, you didn't have a huge swing in your blood sugar. When your body temperature has been at exactly 98.6 degrees Fahrenheit all day. And if you think about it, in your day-to-day life, you're exposed to lots and lots of changes. And as I said, your body is really good at adapting to them. So for changes in light, your pupils can dilate or constrict to let the right amount of light in. For changes in temperature, you can shiver or you can sweat so that your body temperature stays constant. When you eat, your digestive system kicks into action so that your blood sugar stays relatively constant throughout the day. And even when you do things like exercise, you need to maintain the right balance of oxygen and carbon dioxide in your body so you can change your respiration and your heart rate, your breathing and your heart rate. And so since this is such a fundamental thing that happens to you throughout your life, people noticed a really long time ago that your body is really good at adapting to its surroundings. And in 1878, a French scientist named Claude Bernard said, all of the vital mechanisms, however varied they may be, have only one object, that of preserving constant conditions of life in the internal environment. So what he means by preserving constant conditions of life in the internal environment is just that most of your organ systems remain in a pretty constant state, no matter what's going on the outside of your body. And I want you to also notice that he says all the vital mechanisms. He's saying that he thinks everything that your body's doing is basically just to maintain this constant state. And this is mostly true. If you think about it, it's really important to maintain this constant state. Take the example of your body temperature. If your body temperature is at even 103 degrees Fahrenheit, less than 5 degrees above 98.6, that can be really dangerous. If it stays that high for too long, that can make you really, really sick. And also, if you think about a lot of the things that your body does, from sneezing when you're sick to feeling hungry, those are all kind of to maintain your body in this constant state. And in 1929, another scientist named Walter Cannon came up with a word for this, and he called it homeostasis, which literally means same state. And people have noticed that this is so important for life that it's actually part of the definition of life, along with, being able to reproduce and being made up of cells. And so since homeostasis is so important for all of your organ systems, it must also be important for your brain, right? And so that is what I'm going to talk to you about today. But first, we're going to go through a brief introduction about how homeostasis in general works, and then we're going to get to the brain. I'm going to tell you about what your brain, the electricity in your brain is, what is brain activity, how does your brain help you to perceive the world and learn, and then why does that mean that your brain needs to stay balanced? Why does learning make your brain need to maintain homeostasis? Then we'll talk about how your brain stays balanced, and then finally we'll talk about diseases that are related to homeostasis, such as epilepsy and addiction. So first, what is homeostasis? So let's forget about your body for a second and talk about one of the other great homeostasis machines, which is your home heating and air conditioning system. Your house is maintained at a relatively constant temperature, no matter what the weather is outside. And that's done by your thermostat. So you set your thermostat at temperature, and your thermostat knows how far your house is from that temperature, and it can do things like turn on your heater and air conditioner in order to keep your house at the same temperature. So for example, if it's really cold outside, the temperature of your house might want to decrease, but your thermostat notices that. It notices that it's too cold, and it turns on your heater, and that keeps your house at the same temperature. And so your body can also do the same thing, right? So your body is also exposed to changes in temperature from the weather, or it might be exposed to changes in temperature. You know, when you work out, you start feeling really hot. But it's always at 98.6 degrees Fahrenheit, or very close to that. And the thermostat of your body is your brain. So your brain figures out how far your body is from 98.6 degrees, and then it does things like make you sweat or shiver in order to maintain your body temperature very, very close to 98.6 degrees Fahrenheit. And so this guy, Walter Cannon, that came up with the word homeostasis, also gave it a definition. And I want you to think about how this definition relates to this body temperature example I just gave. So what he said was, changes in the surroundings excite internal disturbances of the system. Such disturbances are normally kept within narrow limits because automatic adjustments within the system are brought into action. Okay, so let's look at how this fits with this body temperature example. So changes in the surroundings, that would be like changes in the weather, might make very small changes, internal disturbances in your body temperature, but those are kept within a narrow range by your brain, which makes these automatic adjustments of shivering or sweating. And so just to simplify this a little bit throughout the talk, I'm going to call these external changes, and I'm going to call any changes that occur within the system internal changes. The thing that is measuring how big the change is and controlling these automatic adjustments is going to be the controller, and the controller is going to enact negative feedback. And we're going to call this negative feedback because the feedback always occurs in the opposite direction of the internal change. So what I mean by that is that if you get too hot, you're going to sweat because that cools you down. And if you get too cold, you're going to shiver because that warms you up. All right, so I'll stop here for questions just about the general idea of what homeostasis is. All right, so let's move on. And so now we're going to talk about some basic neuroscience to understand why your brain needs homeostasis. So we're going to come back to this system, right? We have external changes, leads to internal changes, but we don't want them to change too much. So we have a controller that can enact this negative feedback that keeps the internal changes to a minimum. Why does your brain need this? What are the external changes that your brain experiences? Well, your brain is how you perceive the world. And as Descartes said, I think, therefore I am, right? So that means that everything that you experience when you're learning, when you're seeing anything, responding to your world, can cause changes in your brain's activity. So these external changes include anything that you perceive with one of your five senses, anything you see, smell, taste, feel, hear, anything you think, and any movements you make can cause changes in your brain's activity. And so we want to keep brain activity relatively constant for reasons that I will get to in a second. And so we have this controller. We don't actually know what the controller is, but we do know a lot about the negative feedback that the controller enacts. And this is what I'm going to talk about later in the talk. But for now, I just want to come back to this idea of brain activity. The primary reason that your brain needs homeostasis is because it wants to stop runaway brain activity. And so what that means is that you're exposed to lots of things, you're learning, and that all causes activity in your brain. But if you have too much activity in your brain, that's what causes a seizure. Also, overactive parts of your brain can die. But again, it's really important that you have some brain activity, right? Because that is how you're existing in the world. That's how you're perceiving the world. So you can't have too much activity. You can't have too little activity. It has to be just right. It has to be maintained in the same state, in homeostasis. All right, so I keep throwing around this idea of brain activity, but what is brain activity exactly? So you might have heard in the news say that activity in a certain part of your brain is important for some tasks, like maybe activity in your hippocampus is important for memory. But what exactly does that mean? If you zoom into a brain, what you're going to see is a network of neurons. And so this is a drawing by pioneer neuroscientist Ramon Hall, where he's drawing the network of neurons that he sees when he zooms into part of the brain. And all of those little dark spots there are the centers of neurons, and all of the lines you see are neurons reaching out and making connections with each other. And if this part of the brain were active, then all the neurons would be sending lots of signals to each other, and actually this is probably what parts of your brain are doing right now as you're listening to me. And so we're going to learn a little bit more about what this activity means by first just zooming in and talking about what does it mean for one single neuron to be active. Then we'll take one step back, and we'll talk about how activity is passed from one neuron to another and how that relates to how we learn. And then we'll take even a bigger step back, and we'll get back to that picture that we saw in the last slide, and we'll talk a little bit about how activity is passed throughout the brain through your neuronal networks. Okay, but first, what does it mean when I say a single neuron is active? But first, let's just make sure everybody is on the same page about what a neuron is. So you probably know this, but neurons are the cells in your brain that are important for storing and transmitting information. And neurons can come in many different shapes. You can see here, this is a picture, again, a drawing of many different kinds of neurons. See that most of them have this kind of dark, blobby kind of center, and then these extensions that reach out. But another thing you might notice about this picture is that there's different kinds of neurons, right? Some of them have bushy extensions, and some of them have just two single extensions that go opposite directions. And the reason that your neurons are different is because they have different functions. They're activated by specific things in your environment. And so neurons can be activated by, in fact, very, very specific things. And so one example of that is that scientists have found in a 2005 study that there's a neuron in the brain of a human that is activated specifically in response to pictures of Jennifer Aniston's face. It's not activated in response to pictures of Halle Berry's face. It's not activated in response to pictures of Brad Pitt's face. It's actually not even activated in response to pictures of Brad Pitt and Jennifer Aniston. This was when they were a couple. But it's activated specifically in response to pictures of Jennifer Aniston. And so I'm going to give you another simpler example of how neurons can respond to specific things in their environment. So I can actually show you a video of this. And so another experiment showed that one single neuron in the cat's brain is activated in response to a very specific pattern. And so I'm going to show you this video where you're going to be able to see what the cat is seeing, and you're going to hear the activity of just one single neuron in that cat's brain. And so this is a very famous experiment that was done by David Beeble and Torsten Beeble, actually over on the medical school campus in the 60s, and helped them to win the 1981 Olympics. So what you're going to see is this bar of light moving around the screen, and then if you listen, you're going to hear popping sounds. When you hear popping sounds, that one neuron that we're listening to, that means it's active. And you're going to hear that neuron pops more when the bar of light perfectly aligned with these X's. All right, here we go. So here pop, and it pops again. When it goes this way, it's not active, so there's no popping. Not active. And it's going to be active again now. All right, so I hope what you could see is that the neuron is active, it's popping only when the light is aligned with those X's. So that neuron is responding to a very specific thing in the cat's environment, which is the position of that light on the screen. And so those popping sounds that you heard, those are each the neuron being active, as I said, and that's sometimes what you hear called a neuron firing. So what is that firing exactly? Well, I've been talking to you about electricity, right? And so neuronal activity does have to do with electricity. So what is this electricity? Let's move to an example of electricity. Let's talk about how your neuron is like a battery. So a battery has negative ions on one side and positive ions on the other. But those negatively charged ions, those little negatively charged particles and positively charged particles, are separated, so they can't interact with each other. And since it's positive on one side and negative on the other, we say that a battery is polarized. And if we were to take a wire and connect the two sides of a battery, maybe you've done this experiment in school, then the negative ions are going to move through the wire towards the positive ions because nature hates imbalance and because opposites attract. And this is electricity. This movement of ions is electricity. And if you were to plug a light bulb into the wire and the ions then went through the light bulb would light up. to do with a neuron? Well, your neurons are also polarized. If we zoom into a neuron, it has negatively charged molecules on the inside, but positive ions on the outside. And those molecules can't interact with each other because they're separated by what we call the neuronal membrane. And the membrane is just what defines the outside of the neuron. All right, but to get electricity, we need to get ions to move. So how do we get these ions to move? So your neurons all have these channels through the membrane. And the channels are sometimes mostly closed, but they're sometimes open. And when they're open, they let only the positive ions through. So if we open one of these channels, the positive charged molecules are going to move into the neuron. And we see that the neuron is now depolarized, right? Because now the positive and negative aren't separated. So the cool thing about these channels is that they are triggered to open by depolarization itself. So that means that when this part of the neuron becomes depolarized, it's going to trigger these channels to open. And then positive ions are going to come in here. And then this part of the neuron will become depolarized. And then if we move down here, channels here will open, and this part of the neuron will become depolarized. And what you end up with is something like this, where you have the neuron becoming depolarized, and this depolarization moves down the membrane like this. And the other thing that you might notice in this video is that after the depolarization moves along, the positive ions move back outside of the neuron. We're not going to talk about how that happens, but I just want to let you know that it happens in such a way that means that the signal always only moves in one direction. And this movement of this depolarization, this electrical signal in your neurons, is what we call neuronal firing. That's what it means for a neuron to be active. And so when we were listening to that single neuron in the cat's brain, every pop we heard was the neuron doing just that. It was firing every time that light got to the perfect spot on the screen. All right, so now we're going to move on, and we're going to talk about how activity spreads between two neurons. And in that first drawing I showed you how neurons were all reaching out and making all these connections with each other. So if we zoom into one of those connections, what we see is that neurons are connected at places called synapses. And a synapse is basically just where the end of one neuron comes really, really close to the beginning of another neuron. And so when a neuron is activated, neurotransmitters, these small molecules called neurotransmitters, are released into the synapse. And then the second neuron has these receptors that can then receive this signal and activate this second neuron. So when the receptors in the second neuron receive the neurotransmitter signal, the second neuron becomes depolarized, and that can cause it to fire. So what you see is something like this. We have one neuron, fires, the depolarization travels all the way down the neuron to the end. When the depolarization gets to the end, releases neurotransmitters, those are received by the next neuron, and it's depolarized, and then that neuron fires. So this is what happens when activity is successfully passed from one neuron to another. But sometimes what happens instead is that the first neuron is depolarized, it sends its signal, but that signal doesn't quite make the second neuron fire. Instead, the signal makes it all the way to the end of the first neuron, neurotransmitters are released, the second neuron starts to be depolarized, but then that signal kind of fades away. It's not a strong enough signal. And so the reason that sometimes the signal is passed on and sometimes it's not, there's several reasons why this can happen, but one is that there can be different strengths of connections between two neurons. And so if two neurons have a really strong connection between them, then that means that the stimulation, that the signal from the first neuron will be really likely to activate the second neuron. But if the two neurons have a very weak connection between them, then the signal from the first neuron will not be likely to activate the second neuron. And physically, what I mean by a strong connection or a weak connection is that the number of synapses is higher in a strong connection, so the two neurons have more points of connection between each other. And then at those synapses, there are more of these receptors and more of these neurotransmitters are released. And so hopefully, you can see how the strength of connections between neurons can be really important for how activity is passed along in your brain. But I also want to point out that these strengths of connections are not the same throughout your life. In fact, your neurons are always strengthening and weakening their connections between each other, and this is actually how we think that learning happens. And so I just want to give you a very common example of how learning works, and that's the example of Pavlov's dog. So maybe you've heard about this, but if you have a dog like I do, you might notice that when it's time for dinner, your dog knows that it's time for dinner. They get very excited. My dog likes to run around in circles, sit on my lap, and lick my face until I feed her. And Pavlov noticed that when it was time for dinner for his dogs, they drooled. And he used this to his advantage. So he trained his dogs to anticipate dinner just by hearing the sound of a bell. So what he did is he would always ring a bell when it was time for his dogs to eat dinner. And while he was training, he did this many, many times for many, many weeks. And then one day he rang the bell, and the dogs thought about food. And he knew that they were thinking about food because he knows that they drool when they think about food. So he had trained his dogs to think about food just with the sound of the bell. But what's happening in the dogs' brains? Well, we talked about how your neurons are responding to specific things in your environment. And so somewhere in the dog's brains, there are bell neurons and there are food neurons, right? The bell neurons are active in response to the bell, and the food neurons are active in response to food. And before the training, these two neurons have very weak, and somewhere in the brain, right, there's a bell neuron that's connected to a food neuron. That's really important. But before the training, the bell neurons and the food neurons, they have a pretty weak connection between each other, which means that before Pavlov trained his dogs, when he rang the bell neuron would fire, but nothing would happen with the food neuron. But during the training, the bell neuron and the food neuron were activated at the same time, and this happened over and over and over. And we know that just by activating two neurons at the same time, it strengthens their connections between each other. And neuroscientists often say, neurons that fire together, wire together, which just means that neurons that are active at the same time strengthen their connections. And so what that meant for Pavlov's dogs is that after the training, when he rang the bell, it would make the food neuron fire, and the dogs would think about food. So the bell neuron and the food neuron now have a strengthened connection, and this is where the memory of the bell, meaning that food is coming, is stored in the brain, right? So Pavlov's dogs have now formed an association between the bell and the food, and that association is stored in the strengthened connection between bell neurons and food neurons. And so if you think about your own learning, we think a similar phenomenon happens in your brains, right? So if you learn, say, the definition of a word, you're going to strengthen connections between your neurons that are responding to that word and neurons responding to related words that define that word. All right, so now I wanted to take one last step back and briefly talk about how neurons act in a network. So you could think of your neuronal networks kind of like your social network. And so you are like a single neuron that is connected to, in your social network, you're connected to many other people with various strengths of connections between those people, and all of your friends are also connected to many other people. And so neurons work the same way. One neuron is connected to many other neurons and has various strengths of connections between all those neurons. And so if we come back to this drawing that I showed you in the beginning, and we zoom into it a little bit, we can see that neurons are all reaching out and making connections between other neurons. If we look at just one neuron, say, I don't know how well you can see this, but that neuron is reaching out. Any of these lines that it crosses, there's a potential connection. So all of these lines interacting with it here, it could be making connections with all of those neurons. And just to simplify that, I'm going to show it to you like this. And I just want to point out that a neuron can both receive signals from many other neurons. So on this end, it's connected to many other neurons. That's where it receives its signals. And it can send signals to many other neurons. So on this end, that's where it's sending signals. And so the way that this then works in your brain is that when you receive some kind of external change, say this could be the lights from this slide. It's activating, say this is a neuron in your eye. It activates this neuron, and that neuron can spread the signal throughout the brain. It's not going to spread the signal to every neuron that it interacts with, only the ones that it has strong enough connections with. All right, so now I want to summarize what we've talked about with brain activity by taking a little look into what your brain activity is doing right now. So as we talked about, neurons are activated by your surroundings. And so you have neurons right now that are activated by looking at this slide, by hearing my voice, and just by the smell of this room or anything that you're exposed to right now. So neurons are activated by specific things in your environment. And we also talked about how learning can strengthen the connections between two neurons. So right now, the neurons that are activated by this screen and the neurons that are activated by my voice are firing at the same time. And so this makes it so that they are forming a connection with each other. And you're going to remember later that there's an association in your mind now between my voice and these pictures on the slides. And we also talked about how one neuron can communicate with many other neurons. And so this means that when you're looking at this slide, it's not just the neurons in your eyes that are activated, but that activity can spread throughout your brain. Okay, so for you, like this is since you have a relatively healthy brain and you have connections that are of various strengths, when your neurons are activated, you just have a nice modest amount of activity that moves throughout your brain. But we've also been talking about how everything in your environment is making your brain active, and anytime two neurons fire together, their connections are strengthened. So what happens if you're just exposed to lots of things and your connections are always strengthening? Why don't you end up with something like this, where every time you're exposed to anything, every single neuron in your brain is activated? In other words, why don't we end up in this loop where all these external changes in your environment are activating neurons, and then connections are always strengthening, and that makes it so that any external changes then activate more neurons, and this goes on and on and on and on and on. That would be really bad, right? It would be bad because, first of all, how would you ever have a Jennifer Aniston neurons if all of your neurons are activated in response to everything? You'd see Jennifer Aniston's face and it would say, Halle Berry. Or you'd smell pizza and a neuron would say, Jennifer Aniston, right? So your brain can't have this because it wouldn't be able to interpret information. But perhaps more importantly, if you have too much neuronal activity, I told you that that's what causes the seizure. And also, I told you that if you have too much neuronal activity, then your neurons can die. And so it's really important that your brain not be stuck in this loop where it's constantly strengthening its connections and being more and more active. And so that's why your brain needs this homeostasis. It needs this negative feedback in order to balance out all of the activity that you have in your brain from learning and moving and responding and thinking. That has to be balanced out with an appropriate amount of negative feedback. And if you didn't have this negative feedback, your brain wouldn't be able to function, and you probably also wouldn't be able to survive. And so we're going to talk about what that negative feedback is after the break. But first, I'll pause here for questions. Yes?
You said that before the training with the dogs, there was a weak connection between the neuron and the neuron. Why would those two things be connected at all?
That's a good question. So that's kind of a hard question to answer because nobody has actually found this connection between a bell neuron and a food neuron. But we think that since activity is moving throughout your brain, oh, sorry, I'm supposed to repeat the question.
Oh, yeah, and I forgot.
The question is, why would there ever be a connection between a bell neuron and a food neuron at all? So we think that since activity moves throughout your brain, so part of the way that your brain processes information is that you have some sort of external change or change in your environment, and that doesn't just change the neurons in your eyes, but it changes neurons throughout your brain. And so the idea is that somewhere in the brain there is a connection between a neuron that has responded to the bell and a neuron that has responded to the food.
So when you're talking about a single neuron firing, I've recently read that as one could measure brain activity, that it might not be that we're actually measuring just the connection and the electricity, but we're actually measuring a side effect from physical moving, like through a straw, pushing it, and then a side effect of that is electrical conductivity, I guess, and so that's what we're actually doing.
So the question is that you want to know what we're actually measuring when we measure brain activity, right? Is that a question?
Well, I guess I've read that it could be that it's more of pushing through of like physically that makes it rather than more of a muscle rather than just the electricity.
Okay, I'm not sure. Maybe we can talk about this a little bit more afterwards. We know that, so I don't know how to repeat this. The question is, like, is there a physical change in your neurons when they fire, or is it an electrical change? And we know that there are electrical changes because if we take neurons and we grow them in a dish and we put electrodes in them, we can see that there are electrical changes. But depending on the way that brain activity is measured, so if it's measured by fMRI, for example, you're actually measuring changes in blood flow. So there are different ways that neuronal activity is measured, and they are measuring different things, and they're not always measuring exactly. Yes.
In the battery example, the negative ion, in the neuron example, the positive ion.
So
repeat the question.
Oh, sorry. The question is that in the battery example, the negative ions move, but in the neuron example, the positive ions move. So in neurons, the negative ions don't move because the negative ions aren't actually ions, they're negatively charged particles, and they're really big. So they can't fit through the channels. The other reason is because the channels are actually very specific to only, not just positive ions, but certain specific types of positive ions. I don't know why in a battery exactly the negative ions move because I'm not a physicist or electrical engineer. Yes.
There are different kinds of memory. Why do we always remember like a negative memory really much easily? And I guess just like memorizing a number or like a number versus something that happened to us that we remember really well, what.
So his question is, why do we remember negative memories better than positive memories? And so I guess that the reason, I'm not really sure what the reason for that is, but I think it has something to do with the fact that negative memories might be more salient because the stimulus to, like, if you're feeling a shock or something, that's going to cause a bigger stimulus to your brain. It's going to cause a bigger change in activity than if you're just like looking at somebody's telephone number. And so if you have a bigger change to your brain, you're more likely to strengthen connections. Yes?
Yeah, so I want to kind of counter that or ask related phenomenon, like, I guess in the immediate sense, like if you think about a bad memory, you're like, oh man, but at the same time, if you are remembering to a time, even if you're having a bad time, like, it seems to be a pretty common thing that the negative aspects of that memory will fade out, and all you remember is like, oh, we had some good times, whatever. So, it seems like there are situations in which the negative one is like sharper, but the positive ones are more just there. Is that?
I see. So you're asking why negative memories are more, less long-lasting than positive memories?
Well, like, it seems like if something comes up to trigger a negative memory, you'll like have it and it'll be like really strong.
Yeah.
But it seems like at the same time, like they talk about that phenomenon where if you think back to a time, like the bad aspects of that memory sort of fade out. Like you don't remember how you felt miserable at the time.
Yeah. So that's.
Do you want me to try this one?
What?
Do you want me to try this one?
Yeah, you can go for it. So the question. Should I repeat the question? The question is, why are negative memories more like triggerable than like good memories?
Yeah, so those negative memories that you feel like you remember forever, psychologists call them flashbulb memories. And I think what Kelsey said earlier is basically true, that when there's a lot of emotion attached to things, that makes the change in your brain much bigger, and your brain kind of knows to like keep track of all the details. And so then later you have all these rich details that can be triggered by a memory that takes you like right back to that like bad memory, whereas something neutral like a phone number, your brain doesn't feel the same need to like encode everything about what's happening.
Oh, because it's like, this is important to remember everything, so now there's so many things that can break it up.
And that people often like differ in how, I don't know, triggerable these memories are, which is why, some people are more susceptible to PTSD, whereas some people are just able to, not have those memories bother them forever. And yeah, there's a lot of research into, how those cells are actually changing.
How does that affect when you think back to a given episode in time? Even if you were miserable at the time, you don't. And it seems like they say generally, like you don't end up experiencing you don't have that same experience, even if something else. Like when you think back to the time period, you're like, oh,
Yeah, I don't know. I would guess maybe it has something to do with how that memory was formed in the moment, where maybe if you weren't quite as emotional when it happened, or as surprised or something.
I think that's actually a really hard question to answer, because a lot of what we know about memory is from, like animal studies, and we can't be like, hey mouse, think back to a year ago, you were living with your mouse siblings, and were you happy then? So it's much easier to study bad memories, because we can tell when a mouse is scared, for example.
But there is an idea in psychology called the optimism bias, which is the idea that the further back you look in time, the more you only remember the good things.
Yeah.
Which is interesting, has all kinds of interesting implications for how people think about their childhood, like, oh, it was so much better when I was a kid, but also why, more elderly people tend to think of their lives as, yeah, which, maybe that's why it's there, so that, over time you remember the good times. But yeah, there's a whole interesting literature on that.
One thing that's hard, but a little bit more harder subject is looking at older videos on YouTube with World War II civilians experience and deep trauma.
Yeah.
You can watch the reaction on people's faces as they will start talking something lucidly, then they'll start using their memory and get the very vivid details of very traumatic experiences, and their emotions just come out on their faces. So it's one of those things, the memories are around, there's smells, loud bangs, things like that, or even just trying to reflect on something. It's not as visceral as it was when it occurred, but that lucid memory comes out. It's whoa, that's a lot of stuff to remember and carry with you the rest of your life.
Right, so that PTSD kind of symptoms are similar to, it's kind of like a really, really bad memory, right? So it's a super strong stimulus to your brain when that happens, so you remember it really strong. Yes? Yeah.
So, does that mean that? So I've heard that you're the one who probably out, and you're going to off. Does that mean that you're part of your capacity?
So, sorry, do you have more questions? Okay, so the question is, if you have neurons that are responding to specific things in your environment, and you only have a limited number of neurons, which is true. You in most parts of your brain, you don't generate more neurons over time, so you're born with the amount of neurons that you have. If you have neurons that are dedicated to respond to specific things, and you have only a limited number of neurons, you have a limited learning capacity. And so the answer to that is that you don't, but that's because it's not like you have one neuron that, the Jennifer Aniston neuron is kind of, it's not always like that. It's not always just you have one neuron that only responds to Jennifer Aniston. You can also have neurons that are reused that respond to many. And so, when the dogs hear the bell, for example, there are neurons throughout their brain that are responding. There's not just one bell neuron. So there's probably a neuron that's hey, I hear something. And then there's a neuron that's hey, I hear something, and it's this frequency. And then then it's talking to hey, that also has this metally sound. And all of those neurons are communicating with each other. And then there's some neuron somewhere that's hey, that was a bell. And so, yeah, so you don't have a limited learning capacity because your brain can actually reuse. So you have you would hear something else at the same frequency, and you would be reusing that neuron. That's hey, it's this frequency. So it can just form connection by forming connections differently between different things. It can actually, you can learn a lot of different things in just that way by forming connections between things that are, the same neurons with different connections. If that makes sense. Yes.
So now you're talking about use neurons, we use neurons to listen to you and watch the board. We'll go through the same neurons, I'd be listening to my teacher explain something and something that's on the board.
That's a good question. So the question is, if the neurons that you're using to listen to me right now the same as the neurons that you use to listen to your teacher in class? And the answer is that yes, some of them are going to be the same because Kind of like we talked about with the bell example, you have neurons that are just like, I hear something. But not all of the neurons are going to be exactly the same, because remember how we talked about your brain is how you perceive the world, and so if it was exactly the same set of neurons that were activated, you would be having exactly the same experience. But since you're having a different experience, there must be different neurons. Or at least they're activated in different ways. Yes?
So you alluded to a second ago that overactive neuron is linked to seizure. I missed that part, so but is that a similar situation that happens when one's having
Question.
I don't, yeah, do you know the answer to that? Oh, the question is, so when you have seizures, you have overactive neurons in your brain. Is that the same thing that happens when you have a panic attack? You also have overactive neurons in your brain. And I actually don't know the answer to that question. You have abnormal activity in your brain, I assume, during a panic attack, but I don't know that it's as, a seizure is very tied to just overactivity, but I don't know that a panic attack has the same.
Yeah, I think a panic attack is different, but I can't say anything more intelligent than that. It's not quite the same, but it could be, you know, that there are certain connections that are stronger than you want them to be. So the connection between, I don't know, being on a crowded bus and the connection between that and like hyperventilating or something, that might be stronger. And so you have to kind of, you know, train yourself to weaken that connection, would be my guess. All right.
All right,
Let's
great,
take
thank you.
a break for intermission. There are probably, hopefully, some snacks out there if you want to grab them. Also, now is a great time to fill out those surveys to give us some feedback. So there's some pens and extra surveys up here, and we'll come back in 10-ish minutes, 5-ish, 10-ish, I don't know. I'm gonna turn off the
Oh, really?
So, do you want me to grab you?
There's also such a
That was
and then like Katie's was like, I mean, in order to sense I like this. You know, like I just and I actually have these questions on the internet, but it's a little bit harder.
yeah.
Okay.
Is this?
Yes.
So, I could win it.
Yeah. What do you mean? Like cite it in something else? Oh yeah. Okay. So the part of the reason we record it is so to watch later, and also this projector is terrible.
Yeah.
So it takes us a couple days to get. We have a camera back there. We kind of put the slides on top of it, so it's nice. It's like Jeopardy style. The slides are right there, and then it's on our website. There's archives. You can watch any thing. So Yeah, within a few days it should be up there. Yeah. But in general, all of our materials are like free to use as long as you cite us somehow. And we ask that the things that you use them for are also freely available to people. Yeah. It's one of the good things about high quality graphics. Well, I'll tell Kelsey, do you like do anything in this area, or what are you thinking of footnoting it for?
It's really interesting. Yeah. So it seems like it's actually been like,
It's not,
it's
I'm
really
working on social science, how the social sciences.
and that's, I'm not sure.
I see, like presenting research, or okay.
I'm sure, but inside, so they're not sure like which one is the, which one is the causation.
Social science. Yeah.
I guess the
Doing better at integrating
Yeah.
And so
Yeah.
I would say to some of you, I'm doing because now it's not the
Yeah. Oh, cool. Yeah. Perfect. Well, excellent. Yeah, thank you for coming.
I was like, I don't want to say the
I would like something on purpose.
I think this is
Yeah,
I think this is good, actually.
okay. Yeah, I think so too. I feel like this has gotten worse. The
This I
projector?
can see it.
Yeah, but I think you mean like since last year.
It's worse than last week,
Yeah.
I think.
Yeah, I think so too. I think the bulb must be dying. Why is it so yellow? I think you're fine. I mean, I'll check that camera. But the live stream looks fine. Okay.
You gotta be questions that you don't know the answer to.
Yeah, I hear you can answer your own question. Yeah.
I was almost like,
We should have said that.
I like this like molecular neuroscience.
I know. So if I ever get a lecture, you have to come and answer all the questions about cells and I can answer, and I'll be like,
Oh my god, it's funny about this.
I feel like I like to plug my professor's wife is a psychologist who literally wrote a book on Optimism Bias. So I feel like I should like coin the term. I feel like I'm like you're explaining things like slightly different. It's like, I'll just be bad.
Curious are amazing.
Yeah. We just assume it's like a good journey in neurons or something.
I have that's coming up.
All right.
Broadly, and she was like, "Please study the brain. We can't talk about that."
Yeah.
This is
Yeah, that feels like a really big topic.
Yeah, I was like imagining a very like philosophical. I got into it a little bit.
Yeah. He does that. Actually, what time is it? How are we doing?
15 to 9.
Cool. This is a really like good time. I wonder if it's extra credit time.
Ask him questions.
Okay. Cool. I'll just set the camera and then.
Well, I think so. Yeah. It's on. It's on. No, everyone at home.
No, this one is for the camera. Back there. We just work on that. Oh, yeah, now I'm working on the camera. Yeah. They can also see it. Cool. All right. We don't even break. Minutes or... All right. All right. Let's start again. Snacks are probably on by now, but I haven't checked. So we turn on the lights, so hopefully now you guys can see a little bit better, but you have to promise not to fall asleep. And yeah, let's let Kelsey do the second half.
All right. So now we're going to talk about how this homeostasis works in your brain. How does your brain stay balanced? And if you remember this homeostatic system again, you have these external changes, anything you see or think or touch or respond to in your environment, and that can lead to changes in brain activity. But we talked about how we don't want your brain activity to get out of hand. And so we have this negative feedback. And so now this is what we're going to focus on, this negative feedback part of the system. And we need that negative feedback just to remind you to avoid this endless loop of the brain becoming more and more and more active. And so to explain this, I want to come back to this social network analogy, where you're connected to various extents to various different people in your network. And so say this is you, and you have lots of friends. And some of your friends are better friends than other friends. So you have stronger connections with some people than others. And your friends are also connected to each other. And so if you think back to, say, around November of 2016, everybody was talking about politics. And so maybe you were talking about politics too to some people, but then this talking about politics really ramped up. And your connections, political connections with people, got a lot stronger. And so you ended up talking about politics with a lot more people than you would have otherwise. And so you just ended up talking about politics a lot. And maybe that felt kind of overwhelming. And so your neurons have kind of the same thing going on. They also don't like to be talking, or in their case, firing too much. And so they have ways to stop that. And so once again, we're going to talk about this by zooming in, just to talk about how one neuron stops itself from firing too much. Then we're going to talk about how the communication between neurons can change so that neurons stop each other from firing too much. And then we're going to talk about how that works at the level of the neuron. So first, let's just talk about how one neuron can stop itself from firing too much. So we're going to come back to this analogy. Here you are, talking about politics a lot, feeling really overwhelmed. What's something that you could do as an individual person to stop yourself from talking about politics so much? Well, you can make a personal resolution. You could say, I am not going to talk about politics so much. No matter what people say to me, I'm just not going to engage with them about politics. And so your neurons do the same kind of thing. They say, hey, I'm not going to fire unless you give me a really, really, really strong signal. And so we're going to go back to this connection between two neurons again. And if you remember, sometimes when the first neuron fires, it sends a signal to the second neuron and it makes that second neuron fire. But sometimes what happens is that neuron fires and it sends a signal to the second neuron, and that second neuron gets the signal, but it doesn't fire. And so we talked before about how that can be changed, whether or not the second neuron fires, can be changed by the strength of connections between the two neurons. But another way that it can be changed is this blue neuron can do something all by itself without changing any strength of connections in order to keep itself from firing in response to these signals. And so how does that work? So if a neuron becomes overactive, how can it stop itself from being activated? And so if you'll remember, neuron firing happens in response to this depolarization with the channels opening in the membrane and ions moving across the membrane. And so right now we're looking at this part of the neuron here. But if we move off the neuron just a little bit, now we're closer to this big body part of the neuron, you'll notice that there are a lot of channels right at this connection between the body and the long, thin extension. They're right here. So these channels are really, really important for a neuron deciding whether or not it's going to fire in response to a signal. So the reason that they're so important is because when a signal comes along, so this top part of the neuron becomes depolarized. But that's not always enough, as we saw, to get the neuron to fire. It's not always enough for this to cause this depolarization to start this chain reaction of channels opening and depolarization traveling down. So when a neuron does fire, what happens is this signal comes along, it opens these very important channels. This part of the neuron becomes depolarized. That triggers the rest of the neuron to become depolarized, and the signal just keeps moving down the neuron. It continues, that neuron fires. So what could a neuron do to make itself less likely to fire? Well, one thing it could do is just get rid of some of these channels, okay? So the reason that that will make a neuron less likely to fire is because if it's now getting the same stimulus, the same strength of a signal that I showed you in the last slide, that signal is going to come along, but when it gets here, it's only going to have one channel to open. And so it's not going to become very depolarized, and that depolarization is just going to kind of fade away. It's not going to be able to start that reaction, that chain reaction that gets the depolarization to travel down the neuron, and so this neuron is going to be inactive. And so if this neuron wants to be is going to be activated, it needs to have an even stronger signal. It has to have a signal that can travel all the way to these channels now in order to start this chain reaction that causes the neuron to fire. And so we call this change in the number of channels that are in the membrane of the neuron a change in their excitability. And that's because it's making it more difficult for a neuron to be excited or activated. And so if a single neuron is too active, one thing it can do is get rid of some of these channels in the membrane, and that makes it less likely to be activated by a signal. And so this is the first homeostatic negative feedback that we're going to talk about. All right, so now let's talk about how the communication between two neurons can be changed in order to prevent this runaway activity. We're going to come back to this example again. You're talking about politics too much. You're feeling really overwhelmed. Another thing that you could do to stop yourself from talking about politics so much is maybe just weaken your connections with some of your friends. So what you could do, you could set up an email filter that filters out all emails that say have the word Trump or Clinton or Bernie Sanders in them, right? So you're not going to get any emails that have any politicians' names in them. And so neurons do a similar kind of thing. They make it harder for themselves to get the signal from the previous neuron. And so the way that that happens, to figure out how that happens, we have to zoom into this synapse again, right? So this is, again, where neurons are connected. And so if you remember at the synapse, what happens when a neuron is activated is that these small molecules called neurotransmitters are released, and then the neurotransmitters are accepted by the receptors in the second neuron. And so one way to make a synapse weaker, to make it less likely for the second neuron to get the first neuron signal, would be to get rid of some of these receptors. That's going to make it really hard for the first neuron to send a signal to the second neuron because there's not going to be as many things to take in that signal. Another way to do this would be to have the first neuron release fewer neurotransmitters. And so that would make it really hard for the first neuron to send a signal because it can't release as many neurotransmitters, so it's less likely for that signal again to get to the second neuron. And however this happens, weakening the connections can prevent overactivity because it makes it harder for the first neuron to activate the second neuron. So if your brain is becoming too active, another thing that it can do in order to prevent overactivity is to weaken connections between neurons. And then finally, I want to talk about how neuronal networks act to stay balanced. So we're going to come back to this example one more time. So again, feeling overwhelmed, talking about politics too much, one final, maybe slightly strange way that you could stop all of this political talk is to designate a few of your friends to just tell you and other people don't talk about politics so much. And maybe these friends are kind of like the political discussion police. They just quiet down all of the political discussion in your network. Maybe they post pictures of puppies and distract everyone with that. I don't know. But your neurons do a kind of similar thing. So you have neurons in your network that also quiet down the activity of the network. Now they're not distracting other neurons with puppies, but they are preventing the other neurons from being activated. And so the way that this works, to explain how this works, we have to introduce a new type of neuron. So far, we've been talking about just one kind of neuron, and that's an excitatory neuron. So excitatory neurons, when they fire, when they're activated, they tell other neurons, hey, you need to fire too. So when we have an excitatory neuron fire, it tells the next neuron, you fire. Inhibitory neurons do the opposite. When they're activated, they send a signal that says, don't fire. So they stop excitatory neurons from firing. So if we have a neuron like this blue neuron here, that's connected to one excitatory neuron and one inhibitory neuron, and they both fire at the same time, then the inhibitory neuron is going to cancel out the signals from the excitatory neuron. And so that's going to make it so that the excitatory neuron fires less. And so the way that neuronal networks can lower their activity is to increase the influence of these inhibitory neurons. And there's several ways that they can do that. One is just by increasing inhibitory neuron activity. Just have the inhibitory neurons fire more. They're sending more signals. They're saying, be quiet, be quiet, be quiet. And so that makes the excitatory neurons fire less. The other way to do this would be to strengthen connections between excitatory neurons and inhibitory neurons. And that's going to happen in the same way that we talked about before. With two excitatory neurons, we're going to change the number of neurotransmitters and the number of receptors at each synapse. And so what that will do is that even though these inhibitory neurons fire the same amount, this one has a stronger connection than this one. So this one is better able to send its signal. It's better able to stop the excitatory neurons from firing. And so however it's done, increasing this influence of inhibitory neurons in the network can stop the excitatory neurons from firing too much. So if your brain is starting to become overactive, that's another way that it can help to maintain this homeostasis of brain activity. All right, so now if your brain is exposed to too many external changes, too much learning, right, and it's leading to these really large internal changes, then now we know that it can enact this negative feedback. It can lower the excitability of the individual neurons, it can weaken the connections between neurons, and it can increase the influence of inhibitory neurons. And that brings these internal changes back to normal. And so these three forms of negative feedback are what provides your brain with this important balance. So they're balancing out this potential for excessive activity, and this is what's allowing you to interact with your world and learn and remember things without having to deal with any of these consequences of excess activity. All right, and so I'm going to pause here for questions about how homeostasis in your brain works, and then we'll move on to talk about disease. Yes?
Sleeping part of homeostasis.
Yes, but the question is sleeping part of homeostasis? And yes, I actually talk about that a little bit at the end, but yeah, we know that actually a lot of homeostasis, especially this reduction in activity, happens while you're asleep. Yes.
So, you mentioned that let's get rid of and said, sorry. So if you channel, then that limits the firing. So
okay, so the question is, if you're removing channels in the neuron's membrane and you're making it less likely for the first neuron to be able to activate the second neuron, and I said that signal then just kind of goes away, but the person asking the question is saying that, and she knows that energy can't really be destroyed and created, so like what happens to that signal? And so when a neuron gets a signal from another neuron, and as I said, that causes a depolarization of the second neuron, right? So ions are coming in, but there's not a lot of ions coming in, so they're just kind of like dissipating through the neuron. And so if the depolarization isn't strong enough, they don't cause the other channels to open and the signal can't go down the neuron. But what happens to those ions is that they actually just get pumped back out to the outside of the neuron, and so the neuron becomes repolarized. So it's then able to respond to another signal. So if you have a neuron that responds to a signal, right, it's depolarized, and then it has to like fix its polarization so that it can respond to another signal. So what happens to those ions is that they just get pumped back out to the outside of the neuron, and then you have that potential energy, that difference, the polarization, the separation of the positive and negative again, and the energy is, the electricity is kind of like stored in that. Yes?
So the neuron just basically passes like on off like a process, like the ions pass through it, so it returns to its base state after the ions pass through.
Yes. Yeah. Okay. All right, so let's move on, and we're going to talk now about diseases that are related to homeostasis. So first, we're going to talk about epilepsy. So epilepsy occurs when there's problems with your brain's homeostasis. It's not able to keep brain activity balanced. And then we're going to talk about addiction, which is kind of an example of how homeostasis can actually work against us. So first, let's talk about epilepsy. So epilepsy is a disease that's characterized by having lots of seizures. And you have lots of a seizure because this balance between the activity of your brain and the homeostatic negative feedback is off. So there's an imbalance between activity and homeostatic feedback, the negative feedback. And specifically, there's too much activity and too little feedback, and that results in having seizures. And so what's a seizure look like? So we know what seizures look like in people by looking at electroencephalograms, or EEGs. And so you can use EEGs to look at the activity in somebody's brain. And so EEGs are non-invasive, so that means that you don't have to do surgery or anything. You just stick electrodes on the outside of the skull, like you can see with this guy here, and your doctors are able to record the electrical activity of the brain through the skull. And so they're not able to record the activity of individual neurons, right? Because you have billions of neurons in your brain, and there's only like 50 or so electrodes here. But you're able to look at the activity of certain parts of your brain, so certain neuronal networks. And so the results from an EEG study look something like this. So each of these lines here is from a different part of the brain, and all of the ups and downs, the waves and the lines, are changes in brain activity. And so this is a seizure from, an EEG from someone who actually has a seizure during this EEG, and the seizure is right here. So you can see before the seizure, there's not so many ups and downs. Like some of these have some waves, but after the seizure, you can see there's massive changes in brain activity. The brain has become really, really active. And so that's what it means to have a seizure, is that you have massive brain activity, and it often occurs throughout even the entire brain. All right. So this excessive activity can cause symptoms of a seizure. And so if you've ever seen someone have a seizure, either in person or on TV, you might notice that symptoms of a seizure include things like uncontrollable movements, hallucinations like visual aura, which is kind of seeing or flashing things in the visual field, feelings of pins and needles, feelings of confusion, or feelings of fear and deja vu. And so you can imagine this can be very disruptive to a person's life. And also, because we know that overactive neurons die, it can be dangerous to someone's health to have a lot of seizures. So why do we get these symptoms from a seizure? Well, as I said before, usually your brain's responding to your surroundings. So you feel something prickly, and your brain has a neuron or several neurons that fire and say, hey, that was prickly. And that's how you know it was prickly. But what happens in a seizure is that your neurons are activated with little or no external input. And so what that means is that your neurons are kind of acting like the boy who cried wolf here. So they're saying things like, hey, that was prickly, or hey, you need to move now, or hey, you should feel scared, even though there's nothing in the external environment that is sending that message. And so why do some people have seizures? Why do some people have epilepsy? And remember, epilepsy is actually characterized by having many seizures, not just one seizure. But the reasons, there are two main reasons that someone could have epilepsy. The first is that they have experienced some kind of brain injury. This could include like a severe concussion or even a bullet wound to the head. Or someone could be born with epilepsy. They could have some sort of genetic mutation that makes them more likely to have seizures. And for whatever, no matter what reason they have epilepsy, that can cause changes in the brain's activity. It can change the excitability of the brain, and it can change inhibition. And so I'm gonna give you some examples of this. So it turns out that actually many of the genetic mutations in epilepsy are mutations in these channels that we talked about. And so since we talked about how these channels are really important for a neuron's excitability, how likely it is to be activated, you can probably imagine how mutations in these channels could make neurons more excitable or more able to be activated. And so just to give you an example, say that a person has a mutation in a channel that makes it open when it's not supposed to be. So it's open even though there's no signal. So this would result in depolarization, slight depolarization, ions will move in, even though there's no signal from another neuron, from an external source. And so since the neuron is already slightly depolarized, even when there's no signal, it means that when a signal comes along, it's much easier to activate this neuron. So another thing that changes in epilepsy is that inhibition becomes ineffective. And this could either be because the inhibitory neurons could have some sort of mutation in a channel that makes them less excitable, or it could be because strengths between excitatory and inhibitory neurons are different. But however it happens, in a healthy brain, inhibitory neurons are usually able to prevent activity. But in an epileptic brain, they're less able to do this. So this means that in epilepsy, inhibition is lower, excitability is higher. All of these external changes can lead to much bigger internal changes. The neurons are easier to activate. And so you can think about people who have seizures in response to something like flashing light. So strobe lights are a really, really strong external change, even for somebody with a non-epileptic brain. So you can imagine how somebody with epilepsy is exposed to this really strong external change, and that can cause this runaway activity that spreads through the brain. All right, but we've been talking this whole talk about how we have this negative feedback that is really good at keeping our brain activity balanced. So what's going on with this negative feedback in epilepsy? We have these big external changes, but why can't we just lower excitability more, weaken connections more, increase inhibition more, just bump up this negative feedback? And it turns out that actually it seems like in epileptic brains, the brain is trying to do this, but it's not strong enough. It's not able to balance the activity well enough, and so this leads to seizures. All right, so now we're going to move on and we're going to talk about homeostasis and addiction. And as I said before, homeostasis and addiction is related to homeostasis because it's an example of how homeostasis is working against us. But before I talk about homeostasis and addiction, I need to tell you that homeostasis can occur in two different directions. So we've mostly been talking about homeostasis as occurring in response to external changes that cause increases in brain activity. So you can think of this as something like a really bright light. If you were to sit in a room with really bright lights for several days, the activity in the visual part of your brain would increase. But your brain wouldn't like that. So over a period of, probably would take a day or two, it would lower excitability, it would weaken connections, it would increase this inhibition, and your brain activity would return to normal. And the same thing would happen if you were to be in a completely dark cave, the opposite thing would happen, right? If you were to live in a really dark cave for several days, the activity in the visual part of your brain would dramatically decrease. But your brain doesn't like to have too little activity either. That can lead to weakening connections, and then your brain just won't really work as well when you were to come back into the light. So what your brain does is it increases excitability, it strengthens connections, it decreases inhibition. It does the opposite of what it would do if you were in a really bright situation. And so these negative feedback, This kind of negative feedback would then increase your brain activity, again, just bringing it back to normal. And we know that this works because if you take a mouse, scientists have taken a mouse, if you blindfold that mouse, and then if you look at the activity in the visual part of the brain, at the beginning of the experiment, the activity is at some certain level, and then when the blindfold goes on, right, the activity goes down. That's what we would expect, because the visual part of the brain is no longer receiving any input from the external environment. But over a period of a day or two, this activity is going to return back to normal. And as I said before, that's usually a good thing. It's usually good for your brain activity to return to normal. But sometimes there can be a downside to that, and that's addiction. And so I'm sure you've heard of addiction, especially opioid addiction. I'm sure you've heard it's an increasing problem in the last 5 to 10 years, with deaths from opioid overdose increasing dramatically. So what makes these drugs so addictive? Well, drug use is a kind of external change in your environment, which means that when somebody takes drugs, it can change the activity in their brain. And that makes them feel high. And over time, if they keep using the drug, as you might expect, their brain activity changes at first, but if they're using the drug for a long time, their brain is going to want to adapt to the drug being there. And so that causes tolerance, which means that they're going to need to take more drug to get the same effect. They're going to need to take more drug to get the same feeling of high. And also they're going to need to take more drug to get the same change in brain activity as they did when they first took the drug. And so if someone has become tolerant to a drug, and then they all of a sudden stop taking the drug, that's another kind of change in their external environment, which is now going to cause a really big increase or change in their brain activity. And so this is going to cause symptoms of withdrawal. And that's just the discomfort that somebody feels when they've adapted to taking a drug and then they suddenly stop their drug use. And both tolerance and withdrawal are really related to homeostasis, and we're going to get to that in a second. But I just want to point out that this tolerance, having to take more and more drug to feel the same high, and this feelings of withdrawal, which make it really difficult to stop taking a drug, are really part of what makes drug use, what can make drug use be really physically addictive. All right, so we're going to focus on opioid use right now, and so we're going to focus on this one part of the brain called the locus coeruleus. You don't really need to remember what this part of the brain is called. Opioids actually affect many parts of the brain, but we're just going to focus on this one. What I want you to know, though, is that the locus coeruleus is important for controlling alertness. So your feelings of whether you're awake or asleep or how well you're paying attention. And so if we look at the neurons in the locus coeruleus, they have some activity. If somebody takes opiates, the activity of these neurons then is going to go down. This can lead to feeling of a decrease in alertness and feelings of sleepiness, which are all symptoms of opioid use. And as should be familiar to you by now, when the activity in the brain changes, the brain wants to adapt. And so if somebody takes opiates, what happens at first is the activity goes down, but over a period of several days, it's going to come back to normal. And so what this means is that even though the drug is there, they now have normal activity of this part of the brain. And this is what causes tolerance. Oh, yeah. Okay, so this is what causes tolerance. So now the person has to take more drug to get the same decrease in activity and also to get the same high. And this tolerance happens because the brain has adapted, and the brain adapts by increasing the excitability of these neurons. So they're like, hey, you have this drug around, I don't like that, I want to be active, so I'm going to make it easier for me to be active. So all the neurons in this part of the brain then increase their excitability. So then what happens if this person stops taking the drug? Well, the activity in this part of the brain is going to increase, right? And that's because the excitability doesn't change immediately. So the brain's adapted by increasing its excitability, and that's caused this tolerance. But now you get rid of this drug, which is preventing the brain from being active, but the excitability is now really high. So it's really easy to activate this part of the brain. So the activity then increases a lot. And so this is what's going to cause these symptoms of withdrawal. And in the case of opioid use, those are going to be like feelings of jitteriness or inability to sleep, because again, this part of the brain is important for controlling alertness.
and now it's overly active, feeling overly alert to Goldilocks. And so I hope that you can see how brain homeostasis, these changes that we really need in order to maintain a healthy brain, are also part of what makes drugs like heroin so addictive. And so in this way, addiction and epilepsy are kind of opposed. We think about how we might cure epilepsy, we think, hey, how can we boost homeostasis? But when we think of how we might cure addiction, we think, how can we suppress homeostatic feedback? And so you'll remember there's this one part of the homeostatic system that we haven't talked about here. And that's this controller that controls what the negative feedback is doing. And so you can imagine that in order to treat any of these diseases, it would be really helpful to know what this controller is and how it works. And so this is actually still a question of open investigation. It's something that I'm personally very interested in figuring out, but we don't know yet. So that's actually a story for another time. But I want to close with what we do know. So what we know is that right now, your neurons are firing. They're firing in response to seeing these slides, they're firing in response to my voice, and they're strengthening their connections with each other so that you can remember all these things that I told you tomorrow. And this is really amazing. So your brain's ability to process its environment and to learn is really, really amazing, right? And I hope you probably already knew that, but I hope you took that away today too. But the other thing I hope that you realize is that what's also amazing is that all of this external stimulus, all of this learning you're doing, isn't going to cause you to have seizures. And that's because tonight, while you're sleeping, your brain is going to be doing this other really important thing, and that's keeping itself balanced. And so with that, I'd be happy to take any questions.
Drugs or I mean like
Yeah.
all the negativity, like sometimes through your dreams. You know, basically your brain is trying to reconcile or organize what happened that day, and that's why you may have a dream. You know, does that make sense?
Yeah.
Your subconscious is kind of sending you messages. Yeah, your brain's trying to detox, like emotionally or just
Yeah.
you know.
Yeah, so she's asking about the brain detoxing during sleep. And yeah, your brain does like a lot of things during sleep, and I am not an expert on like dreams and what they do for you, but I think changing homeostasis is something that we know happens during sleep. Your brain is, I think we think of it as like trying to make up for all the learning that you did during the day. You don't want to go into your next day with all your connections being too strong and have the potential for overactivity. So yeah, it's kind of like that. Yes.
So like, you know how you said like your body like it's like out of focus because you don't have like the drugs in you?
Yeah.
So like, what if I'm drinking like an excessive amount of water, more than like the body used to? Would the body feel like different because I don't have that same amount?
So you're asking if your body adapts to the amount of water that you drink. Yes. So the way that your brain adapts is actually fairly slow, right? So I'm talking about like over a period of several days, your brain activity changes. So actually, if you don't drink enough water, like if you didn't drink any water for the next 6 hours, I bet you'd feel really thirsty, right? And that's actually just your body adapts really quickly to not having enough water. So it's kind of, that's also homeostasis, but it's of how much water you have in you, because that's actually really important, but it happens on a much shorter time scale. Yes.
What sorts of things can we do to help with that restoration of the neurons overnight?
Okay. So the question, sorry. The question is, what can we do to help your neurons restore their homeostasis overnight? And I don't think that anybody really knows the answer to that yet, because actually that's a fairly recent finding. Just within the last few years, it was shown that this homeostasis happens, this scaling down of your activity happens during sleep. So it's
Get enough sleep.
yeah, getting enough sleep is probably good. Yeah. Yeah, actually, in the study that they did this, they did an experiment in mice. This experiment where they kept the mice awake by like poking them all the time, and they like weren't able to learn as well and stuff.
Yeah, just thinking it would obviously affect your focus, your function the next day. And 3 hours of sleep versus 8, you're probably going to feel different. 5 hours is, so they say that when you sleep, your body's restoring itself, like your immune system is kind of recharging. If you deprive yourself, you're sleep deprived, it's going to affect your immune system. Everything's going to be weakened, or it's not going to function like it's supposed to.
Yeah, I think sleep is important. Yeah. We're learning more about how sleep is important. Yes? Okay, so you're asking if you can, so you have some genetic disposition for your neurons to behave a certain way, or any part of your body to behave a certain way, and you're asking if you can overcome that by like training yourself kind of. I think not in the case of so in the case of like epilepsy, like not really, yeah. So I think it's there are certain genetic disorders. I guess actually there are certain genetic disorders that you can overcome, that you can treat by doing things differently. Like I think, eating certain foods can help with some kinds of genetic disorders. But in general, you can't change what your genetics are. So there, of course, are always ways that you can work to work with your genetics to live a happy life.
Against them, or
Yeah,
to not
that's
against.
yeah. So, but yeah, that you can't just generally you can't change your genetics in a living person usually. But there's some exceptions to that. Maybe you'll hear about them at Daycon if you go.
All right, well, let's thank Kelsey. And as you're getting ready to go, I'll make one final plug for our surveys, get some feedback, what you thought about the event, about Kelsey's presentation. You can fill that out and just leave it up here on the table out by the snacks. We'd really appreciate it. Thanks.